Hello, everybody, and welcome back from lunch. My name is Hilary Vernon, and I am pleased to be moderating this next section at this very exciting scientific updates meeting. The first speaker of this session will be Dr. Head, who is a Pratt Fellow in the lab of Dr. Charles Vendetti at the National Human Genome Research Institute, where she studies molecular mechanisms that drive pathophysiology of methylmalonic acidemia. And Dr. Head's talk is entitled Acyl Modifications in MMA.
Hello, everyone. Thank you for attending my presentation. My name is Dr. Pam Head, and I am a postdoctoral fellow in the Vendetti lab at NHGRI. I'm also a Pratt Fellow for NIGMS at the NIH. Today, I will be discussing my findings on how aberrant post-translational modifications contribute to methylmalonic acidemia, or MMA, pathophysiology, and how these findings have been used to identify and optimize a novel therapeutic target enzyme. Here's my disclosure slide, and I have nothing to disclose. But first, some background. Organic acidemias, or OAs, encompass a large class of genetic metabolic disorders that result from mutations in key mitochondrial enzymes responsible for the breakdown of our dietary intake. One such example, MMA, results from mutations in the enzyme methylmalonyl-CoA mutase, or MMUT, or in the enzymes that generate its necessary cofactor, adenosylcobalamin. MMUT is responsible for the terminal catabolic step in the metabolism of the amino acids isoleucine, threonine, methionine, and valine, as well as for odd-chain fatty acids and cholesterol. Functional MMUT loss results in the accretion of pathway intermediates known collectively as acyl-CoAs, which are subsequently converted to toxic metabolites known as acylic acids. MMA, like other OAs, carries high rates of morbidity and mortality, presenting with a wide array of metabolic perturbations. But it has been previously established that the toxicity from acylic acids cannot account for this complex pathophysiology, such as the development of ketotic hyperglycemia and hyperammonemia. Therefore, my project has been to reassess the MMA disease model and find the true underlying causative molecular mechanisms so that new therapeutic targets might be found. I hypothesized that these acyl groups can be converted, rather than be converted to acylic acid, can be shifted non-enzymatically from their CoA carriers onto nearby protein lysine residues in the alkaline environment of the mitochondria as post-translational modifications, or PTMs. In OAs, such as MMA, an overabundance of acyl-CoAs could result in aberrant hyperacylation of mitochondrial proteins. This aberrant hyperacylation has the potential to disrupt protein function and thereby contribute to metabolic dysfunction and disease manifestation. I used Western blot analysis to examine global acylation marks and was able to confirm aberrant hyperacylation in the forms of both malonylation and propionylation in the hepatic extracts from our lab's primary mouse model of MMA, known as the MCK line. The MCK line is an MMUT knockout line with high expression of MMUT in skeletal and cardiac muscle due to a transgene with a skeletal muscle tissue-specific promoter. This allows for increased survival without eliminating disease pathophysiology in other affected organs, such as the liver and kidneys. Similarly, I found MMA patient liver tissues exhibit the same forms of aberrant hyperacylation. In these Westerns, any protein modified with malonylation or propionylation shows up as a distinct band, and as compared to controls, MMA tissues have significantly greater banding. Also, the pattern of each PTM banding is conserved between mouse and human MMA tissue, indicating a specific set of proteins is consistently modified. I also found the banding for both PTMs was well conserved in our lab's other MMA mouse models. Here, whether MMA was caused by a point mutation in MMUT, loss of MMUT cofactor production, or loss of MMUT expression, the PTM banding patterns remain the same. further demonstrating that these marks are disease-specific and likely have a functional impact on disease phenotype. With that in mind, I will now begin to share my data demonstrating how aberrant hyperacylation contributes directly to the dysregulation of two well-known MMA-altered pathways, the urea cycle and the glycine cleavage system, and therefore to MMA-associated hyperammonemia and ketotic hyperglycinemia, respectively. I will begin with the urea cycle. In this pathway, ammonia is converted to urea, and the first step is catalyzed by carbamyl phosphate synthase 1, or CPS1. In order to function, CPS1 requires the cofactor N-acetylglutamate, or NAG, which is produced by N-acetylglutamate synthase, or NAGs, from glutamate and acetyl-CoA. One theory indicates that in MMA and other OAs, elevated non-acetyl acyl-CoAs, such as propionyl-CoA, outcompete acetyl-CoA for the NAG's binding pocket, leading to the synthesis of alternate N-acyl glutamates, such as N-propionyl glutamate, which are weaker activators of CPS1. Overall, this would lead to decreased CPS1 activity and hyperammonemia. As a result, some patients opt to take carbaglu, a CPS1 activator, in order to bypass the need for NAG. But this medication is very expensive and has unproven efficacy for long-term treatment. And because it does not fully restore CPS1 pathway function, and there must be another cause for hyperammonemia, other than just the reduction of cofactor pools. What is yet to be considered in MMA is the regulation of CPS1 by aberrant PTMs. It is already known that CPS1 activity is hindered by excessive modification with succinyl and glutarate acylation marks, and inhibition is only alleviated by removal of these PTMs by mitochondrial deacylase SIRT5. In fact, deletion of SIRT5 has been shown to result in increased blood ammonia accumulation in mouse models. Unsurprisingly, I found CPS1 to be aberrantly malonylated in MMA hepatic tissues. Specifically, there was increased modification on 15 different CPS1 lysine residues in MMA mouse liver tissue as compared to littermate controls, as found by mass spectrometry analysis, including lysine 1291, the acylation of which has already been associated with downregulated CPS1 activity. To confirm these results in human tissues, I performed an immunoprecipitation assay against CPS1 from MMA and control patient tissues. Here, you can see when purified from MMA tissue, CPS1 exhibits what is likely hypermalonylation when compared to controls. Therefore, it is likely that in part, MMA hyperammonemia can be attributed to dysregulation of CPS1 by aberrant hyperacylation. My next step was to see if aberrant acylation could also contribute to the development of ketotic hyperglycinemia through inhibition of the glycine cleavage system, or GCS. MMA and other OAs often present with ketotic hyperglycinemia, or KH. In fact, several OAs were originally diagnosed as KH until discoveries in the 1960s revealed that it was actually a secondary metabolic perturbation of propionic, methylmalonic, and isovaleric acidemias. However, the cause of KH in these OAs has remained elusive, but today I will present new compelling evidence of the underlying disease mechanism. To begin, the GCS breaks down glycine through the action of 3 enzymes and 1 carrier protein known as H protein. In order to function, H protein must first be modified with lipoic acid on lysine 107 by the enzymes LIPT2 and LIAS, as it is the lipoic acid PTM that carries portions of broken down glycine between the other 3 enzymes of this pathway and keeps the cycle moving. However, I discovered that H protein lacks the necessary lipoic acid PTM when immunopurified from MMA patient liver samples as compared to controls, indicating that GCS function would be greatly reduced. The placement of lipoic acid on lysine is a highly conserved PTM from bacteria to mammals that occurs on a specific metabolic proteins in 4 distinct complexes listed above, including H protein in the GCS. And it was possible that the lipolation placement enzymes LIPT2 and LIAS may be adversely affected in MMA, and therefore the majority of lipolation PTMs would be lost in MMA liver samples. But I found that the majority of these listed enzymes actually had increased lipolation in MMA patient liver tissue samples, likely due to increased mitochondrial mass usually seen in MMA. Only one protein lost lipolation at 18 kilodaltons, the exact molecular weight of H protein, indicating loss of lipolation in MMA was specific to H protein, rather than a result of deficient LIPT2 and LIAS. And because acylation PTMs also occupy lysine residues, I then hypothesized that aberrant acylation could effectively block placement of lipoic acid on H protein, and thus account for the lack of H protein-specific lipolation in MMA. So I then performed a second immunoprecipitation analysis on H protein and found that when immunopurified from MMA human liver tissues, it was indeed aberrantly acylated, but not in controls, and thus established a mechanism for MMA-driven ketotic hyperglycemia through the inactivation of H protein by aberrant acylation. Today, I have focused on 2 pathways dysregulated by aberrant acylation, but it is important to note that many more exist in MMA. I have identified a multitude of MMA aberrantly acylated proteins through mass spectrometry analysis on MMA mouse livers as compared to littermate controls, and have generated protein pathway webs via gene ontology analysis of those acylated protein hits. Highlighted spheres represent pathways I have since validated for aberrant acylation and have found evidence of acylation-driven pathway dysfunction. So if much of the pathophysiology in MMA, and for that matter other OAs, is the result of aberrant acylation, the next question becomes, how do we address it? InMUT replacement alone may be insufficient, especially if InMUT, once re-expressed in this system, could be inactivated by aberrant acylation and slow recovery. Therefore, I believe the answer lies in also developing a deacylase enzyme targeted therapy. In mammals, there exists a family of specialized deacylase enzymes known as the sirtuins. There are 7 sirtuin enzymes, which differ by cellular compartment localization and/or acyl substrate preference. SIRT5 stands out as a prime novel therapeutic target as it is localized to the mitochondria and has an established preference for negatively charged acyl PTMs, such as malonylation and neutral charge acylation PTMs. Furthermore, through in vitro-based studies, I have found that SIRT5 also exhibits activity against propionylation. Here, I set up an in vitro study in which SIRT5 was incubated with propionylated BSA substrate, with and without the necessary sirtuin cofactor NAD+, and with and without sirtuin inhibitor nicotinamide. I also set up an additional tube in which SIRT5 was pre-modified with propionyl and was given NAD+, but not inhibitor nicotinamide. These results demonstrated that SIRT5 could remove propionyl marks, but not if aberrantly acylated, as it would likely be in the MMA mitochondrial environment. So to generate a better therapeutic, I engineered and modified SIRT5 enzyme through site-directed mutagenesis that I will refer to as super SIRT5, which is capable of removing propionyl and other target acylation marks in vitro, even if aberrantly acylated, and that exhibits resistance to sirtuin inhibitor nicotinamide. We have since generated an AAV for this enzyme and carried out a pilot study in the MCK mouse line, and it has yielded promising initial results. But due to time constraints, I will need to end there. I want to thank my lab, our collaborators, and can now take any of your questions.
Thank you very much, Dr. Head. I see one question in the chat that is inquiring if you measured CPS and glycine cleavage enzyme activity to show that the post-translational modification affects enzyme activity.
So in our mouse model, where I didn't see I didn't measure CPS1 activity directly, but rather we looked at blood ammonia levels in the mice, and those that were of MMA mice had much higher ammonia levels than non-MMA mice.
Thank you. And I have a question for you. To what degree do you think that the differences in propionylation and myelination could possibly account for some of the differences between the phenotypes in MMA and PA? For example, we might think that the myelination would actually be decreased in the PA mouse compared to the MMA mouse.
So if I understand your question, it's like the ratio of hyperpropionylation versus myelination, and could that potentially lead to different phenotypes? It's certainly possible, especially with the differences in the PTMs themselves. So propionylation is like a neutral charge, and myelination is negative charge. And so since charge can greatly upset protein activity, propionylation could have a very different effect on one enzyme than, say, myelination would. So it could account for that potentially. Yep.
And sort of along those lines, did you look at any decrease in succinylation? Is that something that you've looked at yet?
Yeah, so I actually explored all various forms of acylation, including succinylation, but it was not altered in MMA at least.
Thank you very much. And I'm looking at the chat. I don't see any other questions right now. Oh, I do see. I see one other question that has come up here. What could be a potential side effect when you express SuperAssert5 by AAV?
So from a preliminary study in mice, I haven't seen any negative effects at this time. It is a mitochondrially localized and isolated sirtuin enzyme. And I believe some studies that have looked at just SIRT5 overexpression don't really see any malignant bad side effects or any sort of side effects at this time. And eventually I want to try and develop more of a activator as opposed to like just the straight AAV on top of that. But at this time, I'm not sure what the negative effects might be. We're still very early in those kind of pilot studies.
Thank you very much. Our next talk is going to be with Dr. Maria Fernandez-Velasco, who is a senior researcher at the Institute of Research in La Paz Hospital in Madrid. She leads several projects related to the involvement of the intracellular calcium mishandling in the progression of cardiovascular diseases such as heart failure. And Dr. Fernandez-Velasco's talk is entitled Mechanisms of Cardiomyopathy in Propionic Acidemia.
Good morning. First of all, I would like to say thank you for organizing to invite me to explain our study related to intracellular mechanisms involved in cardiac disease, so in propionic acidemia. As you well know, propionic acidemia is a rare metabolic disease which results from a deficiency in the mitochondrial enzyme propionic-CoA carboxylase, PCC. Mainly by mutation in PCCA and B that encode both isoforms of this enzyme. This deficiency leads to accumulations of some deleterious metabolic intermediates. There are no cure for propionic acidemia today, and the most healthcare suggests and minimize the protein intake in order to prevent the accumulation of propionic-CoA. Also, L-carnitine supplement is suggesting for this patient, and in some cases, liver transplantation might help to prevent metabolic crisis. The signs and symptoms observed in propionic acidemia includes poor feeding, vomiting, lethargy, hypoglycemia, hyperhammonemia, ketonuria, elevated glycine levels, and also metabolic crisis. Related to the clinical presentation, there are two main forms, the neonatal and the late onset forms. We focused in my talk in the late onset because many cardiac complications was observed in these patients. But also some subjects saw renal, gastrointestinal, immune, and nervous problems. Related with the cardiovascular problem, they saw many cardiac problems in these patients, and is considered a progressive late onset complication and one of the major causes of death in this subject. We can find several impairment in which in cardiac function, for example, systolic dysfunction, some patients saw heart failure with depressed ejection fraction, but also some subjects saw structural remodeling, for example, hypertrophy. In some individuals, there are also disturbances in the heart rhythms, and we can find prolonged QT interval, tachycardia, bradycardia, problems in the conduction system, and ventricular ectopic peaks. It's important to say that all of these impairment and cardiac problems, are closely related with changes at the intracellular calcium handling, especially at the excitation-contraction coupling processes. What is the excitation-contraction coupling? It's the link between the electrical stimulation in the heart and set contractility. And you will very know that the pacemaker activity begins at the sinoatrial node, very specific place in the heart, and the stimulus go across the conduction system, so through his Purkinje, and arrive to the ventricle. What happened in the cardiomyocyte of the ventricle? The electrical stimulation induces a depolarization in the sarcolemma that activates a very specific calcium channel, the LTCC channel. So the activation induces the entry of calcium inside of the cytosol, and this entry of calcium induces the activation of the release of calcium by the sarcoplasmic reticulum through renin receptor. So the activation of calcium induced by LTC channel to calcium release through a renin receptor promote the significantly increase in the intracellular concentration of calcium. This calcium binds to troponin and activate the cell contractility. Once the cell is contracted, they need to relax, and we need to recover the physiological levels of calcium at intracellular level. So the calcium that entry through LTT channel go back to the side of the cell through the sodium-calcium exchanges. And the amount of calcium that comes from sarcoplasmic reticulum come back through the ATPase SERCA2 that is modulated by phospholamban protein. So in our study, we tried to analyze what happened with the cardiac function and with this intracellular mechanism related to calcium in a mice model of propionic acidemia. So to this end, we use hypomorphic PCCA mice that have a mutation A138T that has been firstly described by the group of Michael Berry. And these mice retain almost 9% of the activity of the PCC, but you can see that compared with a wild type, there are significant increases in the activity. These mice also show biochemical and clinical symptoms similar to found to the humans, and they show also elevated cardiac expression of biomarkers of cardiac damage, such as atrial and brain natriuretic peptides. Importantly, these mice also saw mitochondrial dysfunction increases oxidative stress, and all of them contribute to a bioenergetic deficiency. So, using these mice, we performed the following experiment design. First, we analyze the cardiac function by cardiac magnetic resonance, and we analyze protein changes using Western blot and proteomic analysis. Also, the heart was used for obtaining cardiomyocytes, isolated cardiomyocytes, using the Langerdos system. So in this isolated cardiomyocytes, we perform PAT-scan, confocal microscope, and immunofluorescence approach in order to understand intracellular mechanisms and some impairment in this cardiac handling associated with the propionic acidemia. First of all, we analyze what happened with the structural macroscopic and microscopic level. There are cardiac hypertrophy in our mice. For this reason, we analyze the heart rate and also the heart rate tibial length that indicate how the bigger was the heart. And we can see in both groups, wild type and hypomorphic mice with Propionic acidemia, similar level between them. So there are no cardiac hypertrophy at the microscopic level. So what happened at the microscopic level about the size of cardiomyocytes? So to this end, we analyzed the same surface at the capacitance of these cells, and we saw similar level between groups, indicating there are not microscopic hypertrophy. Besides this, we also analyzed what happened with the cardiac function with using cardiac magnetic resonance. You can see here an example of an image of 4 chambers obtained with this technique. You can see the left ventricle, right ventricle, above atrium, and you can see in the video how is the function, the contraction, and the relaxation. So here you can see both example obtaining with these techniques and how is the systolic cycle. It means how is the contraction in both groups. Here you can see in each cycle that the luminal space was lower, but however, this in the wild type group, and we observe the same in the group in the hypermorphic mice. The contraction is not to be complete. It means that the luminal space remains higher at the end of the cycle. So this we can see this effect when we analyze left ventricle and systolic volume. So we saw an increase in this value in the PA group, in the hypermorphic group. Indeed, when we analyze the ejection fraction that indicates the amount of blood leaving the heart with this contraction, we saw a significant decrease in our PA group. This means that our hypomorphic mice saw a clear systolic cardiac dysfunction. And the next question was due to changes or an impairment in cell contractility? And we analyzed the cell shortening that indicates the cell contractility, and we saw this. We saw a decreases values of cell shortening in our in the cardiomyocytes isolated from hypermorphic mice with Propionic acidemia. So the next question was to analyze if this function at the systolic level was related with a calcium mishandling. So the first question was to analyze what happened with the calcium induced, with the calcium release through the LTCC channel. So we analyzed this using PAT-scan, and here we can see the result, the calcium density obtained at each voltage. We can show similar values in both groups. That means there are no changes with the calcium entry into the cell. The next step was to analyze what happened with the calcium release by renin receptors. So to this end, we use also isolated cardiomyocytes, but in this case, loading by fluo3M that allow us to record fluorescences in the same relation with the concentration of calcium at the intracellular level. So we basing our cardiomyocytes at 2 Hz, and we can see here when we establish at the stimulation, there are increase in the fluorescence that then transiently decreases. Here you can see the profile with significant increase of the fluorescence and then go slowly decreases. It's important to analyze the kinetics of these trances because indicating how is recovered calcium by sarcoplasmic reticulum. And we can take an idea on how is CERCA working. So we can see here the result obtained in this experiment. We can see a cardiomyocyte obtained from a wild-type mice, from a PA mice, and also the fluorescent recording obtained in cases. You can see that signal obtained in the PA group seems to be lower than the wild-type. And of course, when we analyze the mean data, the amplitude of these trances was significantly decreases in the PA group and was associated with increases value of the time decay of these trances that indicated that the rate of calcium uptake by the sarcoplasmic reticulum is slower. Something is wrong when the calcium come back to the sarcoplasmic reticulum. So the next step was to analyze what is the amount inside of the sarcoplasmic reticulum in these cases. So to this end, we analyze using caffeine open or the ryanine receptor at the same time, and we can record the amount inside of the sarcoplasmic reticulum. So using the same protocol that before, we record in the fluorescence in the presence of caffeine, and we can see here an example in both cases, and we can see that in the PA group, the fluorescence was significantly lower compared with the wild type, and this we can see at the mean data. So next question was, is CERCA involved in this disturbance? So you have a slow systolic calcium release due to lower SR calcium load. But is the problem the CERCA? CERCA is working worse? Is something wrong with the CERCA? So we analyze by Western blot the expression of CERCA and its main regulator phospholamban in both cases. However, we didn't see any difference between groups in these cases. But we analyze also the CERCA activity PAN by a biochemical assay. And in these cases, we observe a decrease in the function in the PA group. Thus indicated that there are no less or changes at the protein level of CERCA, but however, there are a dysfunction of these ATPs. Because I explained before that these mice harbor oxidative stress, and also in patients, this has been described, we can analyze what happened at the cardiomyocytes. There are more oxidative stress in isolated cardiomyocytes, and if there are something wrong with the oxidative stress, there are involved in changes at the CERCA and can be explained the dysfunction that we observe in these ATPs. So we load our cardiomyocytes with a sensitive dye or frost, H2DCFDA, and we can see that the fluorescence was significantly higher in the PA group, indicated increases oxidative stress. Moreover, when we analyze by proteomic the oxidation and the methionine profiling, we observe significantly increases in both cases in the PA group, indicated an increases oxidation state of CERCA2 that can be explained the dysfunction of these ITPs, that can be explained to problems in the SR calcium load and the systolic deficiency. The next step was to analyze what happened with the diastolic calcium release. It's very important for two reasons. The first one is impairment in diastolic calcium release can be explained at least in part decreases SR calcium load, but also can be involved in some pro-arrhythmogenic calcium event. So we analyzed two forms of diastolic calcium release, as calcium spar, than a spontaneous calcium release. So the calcium sponge you can see here an example in a myocyte. In this case, for diastolic recording, there are no electrical stimulation, so there are quiescent myocytes. And you can see very small calcium release as calcium sponge. And when we analyze the number of the events in both groups, we saw a significant increase in the PA group, indicated increases diastolic release. And we can find the same when we analyze spontaneous calcium transient and calcium waste. There are significantly increased. Here you can see on a sample video how is this spontaneous calcium release in a cardiomyocyte without electrical stimulation. Of course, when you can see this image, you think that maybe this can be a mechanism that induces some arrhythmias. So the last part was to analyze this. What happened with the pro-arrhythmogenic calcium event? We can show during recorded calcium transients some aberrant calcium release. You can see here there is something wrong. And this event was more probably to found in the PA group. When we establish a protocol with serial electrical stimulation, you can see in the wild type regular calcium release, whereas in the PA group, there is something wrong in the calcium release. There are some of spontaneous calcium that can be pro-arrhythmogenic. Of course, the percent of this event was significantly higher in the PA. So finally, we try to understand, is this a normality and pro-arrhythmogenic calcium event at the cellular level have a translation in the mice of the heart rhythm. So we record the electrode in wild type and PA group, and we can see a normal cardiac rhythm found in a wild type mice, and something wrong happened in the PA mice. There are ectopic beats, ectopic ventricular beats that are pro-arrhythmogenic. And the percent of the event was 40% in this group, whereas non-mice saw this event. So in conclusion, our mice with Propionic acidemia saw the depressed cardiac function related to impairment in systolic and also in the low calcium load at the sarcoplasmic reticulum, due to an impairment in CERCA function. Besides this, our mice also saw ectopic ventricular beats related with other increased diastolic calcium leak and pro-arrhythmic calcium event. Finally, I would like to thank all the people that contribute to this work. Firstly, Eva, Richard, and Lourdes Ruiz from CBNCC. They are specialists in the Propionic acidemia studies at the biochemical and metabolic level. They work for a very long time with this disease, and they propose us to understand what happened with the heart. Also, Alejandro Fulgencio, that helped us with experiment. And in Alberto's only institute that sits also Carmen Delgado, Maria Tamaño, Laura Martin, they perform most of the experiment that I showed you. They do the PATS-CLAN, focal microscope experiment, and cardiomagnetic resonance. And the 12 de octubre, Gemma Ruiz and José Alberto Navarro, they perform the electros. Inidi Paz, Almudena Naval, and Marta Gil say help with biochemical analysis. And at CENIC, José Antonio López that perform the proteomics analysis. So thank you for your attention, and I open for questions.
Thank you very much, Dr. Fernandez-Velasco, for that very interesting talk. So I'm looking at some of the questions in the Q&A section, and one of the questions is, what age and sex of mice did you use for this cardiac analysis?
Thank you. But yes, it's a good question. We use male mice at 8 months of age. This age was chosen because it was firstly described to have the metabolic problems, and the group have experience. DJs also saw some problems with the mitochondrial and oxidative stress. So for this reason, we use this age and the gender.
And would you expect to see any differences if you looked in female mice as opposed to male mice?
Can be, because always the estrogens can impair in different way at the cardiac level at least. I'm not sure if there are more probability to find cardiac dysfunction in male than females. I didn't find, because the majority of the study was describing in case report articles. So I don't know, but for example, in heart failure, in many cases, we need to choose males because they are more sensible to find some cardiac problems. But of course, it would be necessary, it would be needed to perform studies in both gender in order to translate our result to both of them. Yes.
Thank you very much. And do any of these specific alterations in sarcoplasmic calcium that you found suggest any specific therapeutics for affected patients?
Yes, I think it's a very good question, because we was very excited at the results. So there are some translational changes at the key protein regulator of calcium at intracellular level that are very close to the cardiac function. So my question was, is in the patient, we can find this behavior in patients. So is patient with cardiac dysfunction to have any post-translational changes in SERCA or in other protein related to calcium handling that can explain the problem? And maybe we can use the therapeutic tools that we use in other cardiac problems, for example, heart failure, or some specific cardiac arrhythmias that is working well for this patient and maybe can also work for propionic acidemia patient. But I think when the clinician, they show this problem, they use the therapeutic tools that normally are used for other cardiac problems.
And one other question from the Q&A group. I know you had mentioned that you saw abnormal oxidative stress affecting the SERCA protein, but this question asks, is the accumulation of other metabolites in PA, could that potentially lead to some of the abnormal calcium release and arrhythmia?
I think it's of course possible, because at the end, for example, also for SERCA and for cell contractility, cardiac function, we need ATPA. So the energetic, when it's compromised, we can find a cardiac dysfunction. And for example, normally the physiological heart, they use the oxidation of fatty acids, sorry. But in some cases, when there are a disturbance, they begin to work the glucolysis in a state of the fatty acids. So in these changes have a deleterious impairment in cardiac function. So of course, some other metabolic problems and the energetic dysfunction can impair the cardiac dysfunction and have a deleterious effect at the heart, of course.
Thank you very much. And thank you very much for a great presentation today.
Thank you.
So our next speaker is Dr. Gerard Berry, who is coming to speak to us today. He's a biochemical geneticist and pediatric endocrinologist. He's the Harvard Levy and director of the Metabolism, Harvard Levy chair, and the director of the Metabolism program at Boston Children's Hospital. He's professor of pediatrics at the Harvard Medical School and director of the Harvard Medical School Biochemical Genetics Training Program. And he is here today to speak to us about liver transplant and cardiomyopathy in propionic acidemia. Thank you very much, Dr. Berry.
Good afternoon. This is Gerard Berry from Boston Children's Hospital. I'm delighted to be here. Many thanks to the organizers for this opportunity to speak with you about liver transplantation and propionic acidemia. Please note none of these disclosures have anything to do with today's presentation. So there are two things that I'd like to accomplish with this visit. One is to review with you the paper that we had published a few years ago on the first instance of a recurrence of cardiomyopathy in patients with propionic acidemia after liver transplantation. And then also we'll talk about the other reports in the literature about when liver transplantation has prevented cardiomyopathy from recurring. I won't speak about liver transplantation per se or about the biochemical mechanisms of cardiomyopathy because they're going to be covered in other slides. So just to get right to the heart of the matter, this presentation is about a male who at the time of death was 19 years old. When he was 2.5 years old, there was the first evidence of a heart murmur. He had his first acute episode of cardiomyopathy at 9 years of age, following implantation of a baclofen pump. And subsequently, a liver transplantation was performed when he was 11 years old. At age 13, he had another acute episode following deep brain stimulation surgery, and he subsequently went into heart failure, and he finally died at 19 years of age. So this is propionic acidemia, and this patient had one of the two mutations that's responsible for the disease process. He had two mutations in the PCCB gene. The patient also had propionyl-CoA carboxylase enzyme activity performed in leukocytes, and which showed markedly reduced activity. This was a full-term baby, and in the newborn period, he was very lethargic, exhibited poor feeding and vomiting and persistent ketosis. He was diagnosed at the age of 5 months and admitted to an outside hospital. He was placed on a low-protein diet and L-carnitine. He began having choreiform movements for which he was treated with multiple medications and which continued all along with dystonia all through adolescence. During infancy, it was felt that he was having seizures. He was started on phenobarbital, and a brain MRI revealed T2-weighted signal enhancement in the basal ganglia bilaterally. Now, I want to point out that this patient was a bit of an unusual presentation. He had more of a cerebral organic acid phenotype with seizures and subacute moving disorder than more of a classic PA presentation. He resembled more someone with glutaric aciduria type 1. He was also evaluated by Dr. Bill Nyan and had been to the facility at the University of California, San Diego, where he was entered in 2 of Bill's studies. Here are some pieces of information supporting the typical laboratory tests that one would find in someone with propionic acidemia, including the presence of urine organic acids such as 3-hydroxypropionate, propionyl glycine, tigloglycine, and methyl citrate. At 2.5 years of age, he received dextromethorphan, and he was on carnitine at 133 milligrams per kilo per day through the UC San Diego group. He was started on growth hormone as part of their research team. He had a cardiovascular exam at one point. He had a regular rate and rhythm. but a grade 2/6 systolic ejection murmur at the left sternal border. This murmur had not previously been noted on earlier exams. An echocardiogram was done, and it was normal. Unfortunately, they had to stop the growth hormones therapy because of insurance issues after it was no longer a part of a research investigation. He was on the typical diets that one would have for someone with propionic acidemia. Please note that these records will be available for anyone to read. So I certainly won't be reading all of these, just some important points. He was first noted to have a left ventricular enlargement at age 7 and possible dysfunction during a surveillance echocardiogram. And then finally, he was diagnosed with dilated cardiomyopathy when he was receiving IV antibiotics for meningitis and developed episodes of hypotension and congestive heart failure. The parents decided they wanted to bring him to an outside neurosurgery department to attempt a trial of intrathecal baclofen pump with the aim to diminish some of his chorea and relieve him of these involuntary movements. He had the pump placed. He reacted quite strongly to a small dose and became extremely hypotonic and fatigued. And upon titration, although his chorea resolved, there was not a much benefit due to the adverse effect of the fatigue and decreased ability to initiate movement. So the intrathecal pump was inserted in the abdomen with the tube running in the spinal canal. And unfortunately, he developed a fever, had to be brought to the emergency room, and he was diagnosed with a staph infection. He was given antibiotics, but the catheter was removed. He developed an abscess, and it was at this point that a murmur was again heard, and now he had a full-blown cardiomyopathy. He had moderately enlarged left ventricle with moderate to severe decrease in global systolic performance with an ejection fraction of 44%. The ejection fraction went down to as low as 25%. There was quite a bit of ethical conferences about how he should be treated. There was a general agreement that a heart transplantation was not a reasonable thing to do. But we decided after discussions with colleagues from around the world that a liver transplantation would probably be the best thing to do. Thankfully, he was able to get almost back to a completely normal function, and he was finally then listed for a liver transplant. And just a brief discussion about liver transplantation and propionic acidemia. There's been quite a few individuals that have had liver transplantation for PA. And this slide shows how cardiomyopathy was reversed after liver transplantation in these individuals. However, please note that in the literature there are also very early reports of individuals who actually died and after transplantation and had cardiomyopathy in that periliver transplantation period. So there's quite a literature with reviews available and with this presentation about the entire scope of transplantation in the literature. Transplantation is considered to be a potentially important treatment to help help eliminate the progression or appearance of cardiomyopathy. This is a picture of this patient's MRI showing abnormal signal within the bilateral putamen. So the transplant was done at 11 years of age. It allowed his 2-methyl citrate level in serum to drop from 60,000 nanomoles per liter to roughly 35,000 in the weeks after transplantation. He was discharged from the hospital, an immunosuppressive regimen. He did have some abnormal liver function tests, possibly due to a drug reaction, but he did develop the complication of PRES after the surgery. There were several manipulations of his immunosuppressive. 12 years of age, he was on his on the diet. He was receiving tacrolimus as the main agent, the family decided that they want to try once again to try to see if they could prevent the terrible movement disorder. So he had a deep brain stimulation surgery at another hospital, and unfortunately, following this, he developed a fever and then eventually pulmonary edema, and had a return of the cardiomyopathy. When he was 18 years old, he now had chronic kidney disease, thought to be largely due to the immunosuppressive surgery, but perhaps also with the combination of the intrinsic disease due to PA. The cardiomyopathy was worsened. He was getting oxygen at night, and now he's the New York Heart Association Class 3 individual. And here's a picture of his chest indicating the edema and the severe scoliosis during his last admission to Boston Children's Hospital. So he was admitted to the intensive care unit at that time, and it was thought to be fluid overload or possible aspiration pneumonia. He was given diuretics. He had severe left ventricular dysfunction. He had runs of non-sustained ventricular tachycardia. He was placed on a lidocaine infusion. He became increasingly more difficult to diurese, and was in florid heart failure. As he continued to have decompensated heart failure in his last few days of life, he became more hypotensive despite vasopressor support, despite efforts to maximize medical therapy with diuresis and afterload reduction, inotropy, and non-invasive positive pressure ventilation. He continued to decompensate, and after the ethical team and parents met, there was a decision to redirect care, and he died on his birthday at age 19 years of age. So for your review, there's a substantial amount of references that contain all the information about liver transplantation, when liver transplantation has been associated with death in the newborn period and cardiomyopathy, as well as the relatively large number of successes where liver transplantation has helped to eliminate or retard the development of cardiomyopathy. Many people to thank who helped with the care of this patient as well as in the generation of the publication. And thank you very much for your attention.
Thank you very much for that presentation, Dr. Berry. Let's see. So to what extent do you think that intercurrent stressors such as infection in this patient exacerbated his cardiomyopathy?
I think that was the major reason for the cardiomyopathy. And I also want to say we're still very optimistic about the beneficial effects of liver transplantation in patients with severe propionic acidemia. So much so that I think it's not unreasonable for people with a severe phenotype to actually have transplantation very, very early in life. But I think this seems to be a recurrent theme in patients with PA, how cardiomyopathy can develop with a stressor, sometimes even in the absence of acidemia or hyperammonemia. So I think both of those manipulations that resulted in a medical problem brought out the cardiomyopathy.
Thank you. There's a question in the Q&A asking if prealbumin levels correlated with cardiomyopathy.
I would say no, because of course this patient, there was a number of individuals who had cared for him and had used medical food in the past, and a lot of it, maybe more so than I would have, but most of the time the prealbumin level was normal, but I can't answer it directly because it's not been looked at in a systematic way.
Yeah. Is your impression that some of the effects of liver transplant could be sort of metabolic stabilization to avoid these events that exacerbate cardiomyopathy?
I think so. Yes. I mean, we keep learning over and over again that the cardiomyopathy of patients with small molecule diseases has a remarkable ability to disappear. To disappear with time, and that actually happened with our patient. Our cardiac team, which at one point had given him maybe 5 months to live, he was actually back almost to normal, and didn't have a large amount of dysrhythmias, during his entire course.
And there's another question in the chat that says, from Dr. Kaler, one of our earliest PA patients at Duke on carnitine since age 3 was doing wonderfully, but succumbed suddenly to CMYO and arrhythmias in the aftermath of pneumonia. I'm wondering about the role of inflammation elsewhere triggering CMYO.
Well, that's a good thought. One of the reasons why I belabored the discussion today, I wanted everyone to hear about all the details, because we kept asking ourselves, why is this patient different than the rest? In being the first one to be successfully transplanted and then have a recurrence of cardiomyopathy, but we couldn't identify something. And I think giving back to what Steve said, that I think there's many different stressors that can bring it on. It's not all the same one.
Thank you very much. Let's see. Another question has popped up. In a situation where the heart is involved to a severe extent, what are your thoughts of using an MRI to look for cardiofibrosis? And could that help dictate which patients may benefit from liver transplant alone versus liver heart transplant?
Well, I think this is an area where there deserves a lot of further study to try to look at when does the remodeling process get to be so severe that you've reached the point of no return? And I think this also needs to be studied in a more careful, systematic way. Yeah.
Wonderful. Thank you very much, Dr. Berry.
Okay, thank you. Real pleasure.
Now we're going to hear from Dr. Stephanie Grünewald. Dr. Grünewald is a member of the consultant team of the Inherited Metabolic Diseases at Great Ormond Street. She's also a senior lecturer at the Institute of Child Health, and she's going to speak to us today about liver neoplasms in organic acidemias, an emerging complication. Thank you, Dr. Grünewald.
Good afternoon from the UK, and many thanks for the organizer to invite me to speak at this interesting meeting about emerging liver pathology in organic acidemias. These are my disclosures. Just very briefly as an introduction, organic acidemias are multi-system diseases. They do have common long-term complications that are affecting various organs, including kidney and heart, pancreas, central nervous system, vision, hearing. And as expression levels of the respective enzymes of the different organic acidemias are particularly high in or highly expressed in liver, the liver plays an essential role. I'm concentrating today on liver pathology and an emergent complication of liver neoplasms observed in patients with organic acidemias. Despite its key role, hepatic complication has been scarcely described, though in 2018, Imbat et al. Raised awareness of long-term liver disease in MMA and PA patients. They included 28 patients in a longitudinal study and reported that half of them had signs of liver abnormalities, either on biochemical testing and or imaging and or biopsies. As shown on the right-hand side on the graph, they also saw longitudinal elevation of alpha-fetoprotein seen as a marker of increasing toxic effect on the liver over time. They also reported on abnormal respiratory chain enzyme activities in 2 liver samples they investigated. Although the findings on liver abnormalities cross investigations were only mild to moderate, in 3 or 4 biopsies, they saw fibrosis and cirrhosis. And they concluded that long-term monitoring of PA and MMA patients should include liver evaluations. Around the time when this was going public, we saw the following MMA case. It was a baby boy and he was and is the second child of his consanguineous parents. He has a 4-year older brother diagnosed as a neonate with a mutase deficiency, and the parents have opted out to have prenatal testing. This younger brother was found to have polycystic kidneys on antenatal scans. The baby was prospectively treated, and we started him on low-protein diet and carnitine, and for a short time he received ammonia scavenger drugs. His diagnosis of mutase deficiency was also confirmed, and both siblings are homozygous for a mutation predicted to introduce a termination codon. There were early difficulties to tolerate and build up oral feeds, and he also suffered from recurrent urinary tract infections. Around the age of 3 months, whilst having unexplained episodic hyperlactidemia, on routine blood monitoring, abnormal liver enzymes were reported. Particularly, he was seen to have high gamma-GT and alkaline phosphatase. He also had high alpha-fetoprotein levels, raising up to more than 20,000 nanogram per ml. This was followed up by a liver ultrasound showing a round heterogeneous lesion in the 7th segment. This was seen on this ultrasound, and further imaging, which also depicts the multicystic dysplastic right kidney. Liver transplantation was thought to be the primary option for both diagnoses, the MMA and the liver neoplasm. He was listed, and at 6 months of age received the left liver cadaveric liver transplant, and the detailed examination of the explanted tissue is seen here. So in panel A, you see the hepatoblastoma part of his tumor comprising embryonal but as well fetal compounds. And you see the positive nuclear beta-catenin expression in the embryonal component, which is not seen in the fetal component. And then in panel C, you see the part of the tumor that was diagnosed or was reported as hepatocellular carcinoma. And you see the well-differentiated cells with some steatosis and stromal invasion. The patient was liver transplanted then at the age of 6 months, as I said, and he's now 3.5 years old and 36 months after liver transplant, doing really well. There were already 2 reports in literature about MMA and liver neoplasm. Cousins et al. Reported back in 2008 the first patient with liver tumor in MMA, a mutase patient who had early renal failure and then received a liver transplant at the age of about 10 years. A year later, his condition really worsened dramatically, and he had rapidly increasing hepatomegaly. His fetoprotein levels went very high, and he died 3 weeks later. And on post-mortem finding, he was shown to have a hepatoblastoma resembling adult hepatocellular carcinoma. The impression of the authors was that he had impaired OXPHOS because that was measured in the native kidney, and possibly a toxicity from his immunosuppressive treatment as the reason why he developed this tumor. And then Chan et al. Reported in 2015 another MUT0 patient. He developed at the age of 19 months hepatomegaly, had alpha-fetoprotein levels of above 500,000, and the CT showed a large mass confirmed that it was a hepatoblastoma. The tumor was so big that it was unresectable. Then he had several neoadjuvant chemotherapy, multiple complications, and finally received a domino liver kidney transplant at the age of 2 years and 3 months. And at the report, he was 7 years old, doing quite well. And the authors favored a role of increased reactive oxygen species leading to his tumor development. We then brought the 3 cases to a workshop at an EMG meeting in Hamburg, and we were then notified that there were 2 more cases. So what I call the Swiss case, this young person was diagnosed as a neonate, had a relatively mild course, but then at the age of 23 presented with bilateral visual and partial hearing loss. At the age of 31, she became acutely unwell and was then found to have a lesion in her liver, later confirmed as hepatocellular carcinoma, which was resected. A year later, she died of an unexpected complication of acute hemorrhagic pancreatitis. And then the last patient is another UK case, this time a CBLV patient, diagnosed at 9 months of age with chronic renal failure at 9 years, commenced dialysis at 16 years, and those parents opted out for transplantation. She developed a severe basal ganglia stroke, and at 22 years was admitted with a severe metabolic decompensation. An ultrasound showed a small liver lesion, but then she died very quickly, and only on post-mortem sample, it was confirmed that she also had a hepatocellular carcinoma. So brief summary of the reported cases that we have. They are all MMA cases, 4 with MUT0 and 1 CBL subtype. There is a mixture of liver neoplasm with hepatoblastoma in 1 patient, hepatocellular carcinoma in 2 patients, and the mix of hepatoblastoma and hepatocellular carcinoma in 2 patients. And this concomitant finding of 2 different tumor entities is particularly intriguing, as their pathomechanisms are meant to be fundamentally different, with the hepatoblastoma emerging from fetal liver tissue, whereas the hepatocellular carcinoma is usually a malignant transformation of well-differentiated hepatocytes. So we came up with this little graph reflecting of the various metabolic disturbances potentially promoting oncogenesis in organic acidemias, and many of these causes and relationships have already been supported by literature. So first of all, we have the mitochondrial dysfunction. There are several reasons. The accumulation of Propionic-CoA, which then inhibits the PDH and the succinate-CoA ligase, which then has an effect on the production and the maintenance of mitochondrial DNA. Then there is the disruption of the TCA cycle because you have the deficiency of succinate-CoA. And then there's an excessive formation of 2-methyl citrate, which then inhibits further enzymes of the TCA cycle. And second, we have the mitochondrial dysfunction, which increases the reactive oxygen species. And the increased oxidative stress can obviously lead to DNA damage and pro-oncogenic signaling. And then third, the accumulation of Propionic-CoA results in dysregulated histone acetylation. And more recently, the group of Amsterdam has shown that in PA patients, there's increased proteinylation throughout the cell over and above just mentioning the histones. And this has potentially an effect of gene expression. And then fourth, we speculate that the TCA impairment really leads as well to some mitochondrial dysfunction by increasing oncometabolites. And oncometabolites in organic acidemias have been described before. Fumarate, obviously in fumarate hydratase deficiency, succinate in succinate dehydrogenase deficiency, and 2-hydroxyglutarate in D2-hydroxyglutaric aciduria. So far, we are not aware of a particular oncometabolite in MMA or PA, but looking at the next case is interesting because this is a child with MMA and a renal cell carcinoma, and pediatric renal cell carcinomas are extremely rare, and they are thought to appear sporadic, but there are a number of syndromes and as well gene diagnoses that are associated with RCCs. Our patient has shown a germline mutation in the tuberous sclerosis complex TC2 gene. This 6-year-old patient was actually asked to be seen by oncologists prior to liver and kidney transplant because of persistent hypercalcemia and increasing parathormone-related peptide. He was then found to have some abnormal PET uptake in his left kidney and had a partial nephrectomy, which showed a renal cell carcinoma. And then on tumor whole exome sequencing, 2 inactivating somatic mutations were seen on the TSC2 gene. And this is leading to a disruption in the mTORC1 upregulation, which leads to an accumulation of fumarate, which is a non-oncometabolite. So there are actually even more pathomechanisms that might play a role, and I'm not talking in more detail about unbalanced autophagy or increased autolysosomal biogenesis, but they can both result in an impaired quality control, thus facilitating organelle aging. Now, how do we take these things in account, and what kind of research would be helpful to further identify the possible pathomechanisms for organic acidemia related to oncogenesis? Well, ideally, we would obviously have a metabolomic proteomic profile in microdissected tissue samples. We certainly will use the tissue of our patient for whole exome sequencing of germline and somatic mutations, and they're looking at single-cell transcriptomics in healthy liver tissue and his tumor compartments, hoping that we get some more insight. What does all of this mean for overall the surveillance of our patients? We think, as Imbad said as well, we need regular monitoring of liver function tests, possible serial alpha-fetoprotein levels, which is a good marker for hepatoblastoma. There might be other markers that might be taken in consideration for the future. FGF21 is a predicted biomarker for disease progression, and possibly we really need serial imaging, and if any need is to do more in-depth studies and escalating to biopsies as appropriate. And this is certainly important when we consider transplantation for our patients. Prior to any transplant, we need to identify exactly where the liver function sits and where possible problems are already emerging. And I would have a low threshold to consider liver biopsies prior to transplant. And for the post-transplant time, we need to take in consideration the hepatotoxicity of immunosuppressants. And it was interesting to see a paper last year where the Japanese had a liver transplanted 106 pediatric patients with inborn errors of metabolism. Of those, 36 patients had shown liver dysfunction after transplant needing biopsies. And in 10 out of 11 MMA patients, the biopsies showed diffuse macro and microvesicular steatosis. Now, all these patients had living-related donors, so carrier livers being transplanted, and it just shows the possible increased vulnerability of these organs. Looking at innovative therapeutic approaches, we need to minimize and exclude risk of insertional mutagenesis in adeno-associated viral gene delivery. I think that preclinical studies need absolutely to minimize genotoxicity, and the trial protocols should take into account the intrinsic risk of liver toxicity and possible risk of oncogenesis in organic acidemia. And we need, obviously, long-term safety and efficacy of gene therapies of PA and MMA, and systemic mRNA therapies. They do need further attention. So in summary, we recommend regular monitoring of liver complications, of possible liver complications, organic aciduria, and we would say to evaluate added liver toxicity, oncogenicity in forthcoming new therapies in cell and animal models, and implement stringent surveillance in trial protocols. With that, I thank you for your attention.
Thank you very much, Dr. Grünewald, for that very interesting presentation. So we have a question from one of our attendees, Dr. Cedarbaum, who asks, hepatocellular carcinoma seems to be emerging as a complication of a larger number of inborn errors. And this is also true of urea cycle disorders. Do you think that the mechanism may be more general and due to inflammatory mechanisms, perhaps the final common pathway of a number of pathogenic pathways?
It's a comprehensive question, and I do agree with all areas mentioned needs to be explored. If I could see instantly a similar pathomechanism, then I would really think we have to do a lot of metabolomics on these tissues. My prediction is, apart from the other pathomechanism that we discussed, we will probably find disease-specific oncometabolites for the different diseases.
Thank you. And you certainly talked about these very illuminating cases in MMA. Have you seen any similar tumor profiles in propionic acidemia?
So I haven't personally seen that. My worry is as much for the propionic acidemia patients as it is actually for the MMA patient. And hence the summary says that we should evaluate very carefully the liver function for both diagnoses and actually even for the whole group of organic acidemias.
Thank you very much. I don't see any other open questions. Do you have any sort of closing remarks or any parting remarks for us to take away?
I have. Thank you very much for a very interesting part of the meeting already. Looking forward to the panel discussion later.
Wonderful. So it looks like we're going to head off to break a little bit early. And thank you very much to all the speakers on the panel this afternoon for some really very illuminating talks.
Thanks, Hilary.